The average human heart beats roughly 100,000 times a day, maintaining a continuous pressurized circuit of oxygenated blood. When that muscle suddenly seizes and stops pumping, a strict biological clock begins ticking. In medicine, clinical death begins the moment the heart stops beating and the lungs stop breathing. Because this state is defined by the cessation of circulation and respiration, rapid interventions like CPR and defibrillation can sometimes reverse it. This split-screen matrix highlights a critical distinction. Clinical death is marked by circulatory failure, while biological death is the irreversible cessation of all brain function. In sudden cardiac arrest, clinical death acts as the trigger, initiating a rapid, irreversible countdown toward biological death. The transition follows a strictly defined 60-second cascade of ischemic and biochemical failures. Over the course of a single minute, the failure of blood flow causes the collapse of three parallel systems: the systemic vascular network, the neurological pathways, and the internal cellular machinery. Tracking the exact physiology of these 60 seconds reveals the specific mechanisms that cause the window for resuscitation to vanish so quickly. Our timeline begins at 0 seconds, the precise moment the heart stops its effective pumping. This timeline tracks the simultaneous collapse across 3 levels of the body. Without a pulse, arterial blood pressure drops vertically to 0 almost instantly. Because systemic pressure has collapsed, cerebral blood flow halts. The cerebral cortex enters a hypoxic state, starved of fresh blood. Observers may notice irregular gasping movements in these initial seconds. These are agonal gasps, a physical brainstem reflex that fails to bring air into the lungs or provide systemic oxygen. The brain lacks the capacity to store oxygen. Neurons require a constant supply of energy to maintain their electrical charge, and they consume their residual oxygen supply within seconds of perfusion stopping. Medical literature indicates total loss of conscious awareness occurs within a biological window of 6 to 20 seconds after blood flow stops. By the 10-second mark, the loss of systemic blood pressure ensures that the patient's subjective conscious experience has ended. As the timeline moves into the 10 to 30-second window, the failure spreads from the higher reasoning centers of the cortex into the deeper structures of the brainstem. Within the neural column, organized electrical pulses governing the brainstem break down. Synaptic firing becomes chaotic and rapidly extinguishes. This brainstem shutdown results in primary flaccidity. As the brain stops sending baseline signals to maintain tension, the body undergoes a sudden global loss of muscle tone. Distinct clinical markers define this stage. The jaw drops and sphincters relax. Pupils stop reacting to light as the eyes lose their moisture. During this shutdown, instruments occasionally record transient electrical spikes in the dying brain, specifically bursts of high-frequency gamma waves. This EEG readout separates normal function from distress. The top line shows rhythmic patterns of a conscious brain. The bottom line captures disorganized spikes of an anoxic surge. These are disorganized responses to oxygen starvation, lacking structural integrity to support complex thoughts. Visible twitches or electrical brain spikes in this window are mechanical anoxic reflexes, rather than indicators of lingering awareness. To understand the final half of the first minute, we must look inside the liquid environment of a single cell. Entering the 30 to 60-second window, all residual traces of intracellular oxygen are depleted. In the cellular column, the lack of oxygen forces a metabolic shift. The cellular machinery pivots from aerobic metabolism to anaerobic glycolysis. This backup system produces a toxic byproduct, intracellular lactate, which begins to accumulate rapidly. Lactate accumulation causes the cellular pH to plummet, creating a highly acidic environment known as acidosis. As ATP zeroes out, membrane pumps halt. Sodium and calcium flood in, causing the cell to massively swell. This acidic environment ruptures internal lysosomes. Autolysis is triggered. Released digestive enzymes rapidly dissolve the cell's internal structures from the inside out. While visible decomposition takes days to manifest, microscopic self-digestion begins before the first minute is over. As the biological clock crosses the 60-second mark, the body completes its initial transition. This summary matrix contrasts the body's outward stillness with the catastrophic microscopic breakdown occurring at the 60-second mark. Macroscopically, the body is unconscious and flaccid. Microscopically, it is acidotic and actively autolyzing. These physiological facts clarify several misconceptions. Brainstem auditory pathways fail alongside every other reflex within seconds of blood flow stopping, ending the capacity to process sound. Post-mortem hair and nail growth is likewise an illusion. Because cellular division has ceased, the apparent growth is caused by the dehydration and retraction of surrounding skin tissue. Within 60 seconds, the human body transitions from a cohesive, oxygen-driven entity into a collection of rapidly degrading cellular environments. Consciousness vanishes in seconds, followed immediately by the quiet biology of breakdown.